Good morning. It is Monday, August 24th, and this recording is going to be in lieu of me doing live lectures. I'm going to try this as an experiment, and what I want you to do is while you're watching this lecture, I've enabled the chat feature so you can take little notes if you have questions for me in the chat. They will be seen by everyone. So this is just regarding the lecture, of course. And when you're done watching this, we will, I'll answer those live. Okay. And I'll be having some demonstrations as well. Okay. So we've talked about atoms, elements, hydrogen, oxygen, nitrogen, carbon. Those simple elements come together in simple molecules, which we call monomers. Those monomers are joined together by dehydration synthesis into polymers. Okay. And those polymers, another name for polymers is macromolecules. And these are the four major macromolecules of life: lipids, nucleic acids, carbohydrates, and proteins. Today, we're going to focus on carbohydrates. And then, of course, we will do the other ones as we progress through the unit. All right, so without further ado, you should recognize this diagram here. This is the glucose, uh, glucose diagram. What am I trying to say? An organic chemistry diagram of the structure of a glucose molecule. The three elements are carbon, hydrogen, and oxygen. Remember last week when I had those monomers and I joined them, the paper ones, by linking together the H from the hydrogen from one end of the carbon of the monomer to the OH from another end, and a molecule of water was produced, and we were linking those two monomers together. So this should look familiar for you. The the chemical formula is C6H12O6. Know it, love it, tattoo it on. No, just kidding. Okay. So we call carbohydrates is actually the nutritional term. The biochemistry term is saccharides. And saccharides literally means sugar. Now we know that the monomer is a monosaccharide. Okay. And there are three monosaccharides you need to know: glucose, galactose, and fructose. Hopefully, one thing you will notice about all three of those examples that they all end in ose. So if you see a word in biochemistry that ends with ose, you're probably talking about a saccharide or a carbohydrate. Now, all three of these have very similar chemical formulas. They actually all are C6H12O6. So how do you know which one is which? Well, they are laid out, they are organized a little bit differently. Glucose and galactose are um, are both formed in hexose rings, and they may look almost identical. But if you notice, on glucose, the hydrogen is above this carbon and the OH is below. Whereas on galactose, the OH is above this carbon and the H is below. Subtle difference, but it makes a difference in how those monomers, those monosaccharides, form polymers, the bonds that they form. Now, fructose, as you can see, is a pentose shape. It still has 6 carbons. Let's count. There's 1 on this corner, 2, 3, 4, 5, and then 6. And it's still H12 and it's still O6. But in this case, the oxygen that is bonded here is not bonded on the corner, it's bonded on the top. All right, so those are the 3 monosaccharides you need to know. Now, when 2 monosaccharides come together, we get a disaccharide. We don't call it we call it a monomer, and then 2 monomers together make a dimer. And there are 3 possible combinations. If it's 2 glucose monomers coming together, that becomes a disaccharide called maltose. And that is, I mean, literally the only thing I know of that maltose is in is it's a malt, like in beer, malt beer. Glucose plus fructose is sucrose, which is table sugar. So here you see the hexose glucose molecule and the pentose fructose molecule, and together they form a bond here in the middle, which is called a glycosidic bond. Glyco means sugar, okay? Glycosidic bond, and that is table sugar. Fructose comes from fruit, glucose comes from other sugar producing plants, and then together we get that sucrose. So that's what you put in your tea, that's what you put in your coffee, that's what you sprinkle on your sugar. Sprinkle on your cereal. So that's that. Now, if we take a glucose and a galactose, that gives us lactose. And I'm sure you've heard of that word before because whenever we hear lactose, we always think lactose intolerant. Well, what does lactose intolerant mean? Lactose intolerant means that someone who drinks the milk does not have the enzymes to break down to perform hydrolysis on that disaccharide and cannot break the dimer into two monomers. So, in other words, you don't get the nutrition from that milk sugar because you can't break it down. You don't have the enzyme that would snip those two disaccharides apart. So, it kind of collects in your gut and you get stomach aches and diarrhea and all of that. No fun. Okay. Now, fortunately, there are enzymes that you can take. Lactate is one, and we're going to be studying enzymes later this week that can help do that if you don't have the enzymes in your body. Now, once we get long chains of monosaccharides, that becomes a polysaccharide. As we learned last week, poly means many. Here you can see that starch. This is what we eat a lot of in our French fries, potato chips, corn, all those things. Starch is a bunch of glucose monomers, about 300 to 600 glucose monomers long, and we call it specifically amylose. Again, there's that ose word. Technically, starch, of course, doesn't have an ose, but amylose is, and that's just one monomer bonded to another by dehydration synthesis. And what they form are these glycosidic bonds. So that's starch. Now, another type of glucose monomer bonded to glucose monomer that we find in plants is cellulose. And we're going to talk about that in just a minute. But I know it's probably not very obvious to you, but here the oxygen is bonded in this like a V shape with the oxygen at the base of the V, whereas in cellulose it's bonded this way. That's going to make a difference in just a second. Now, animals also have some starch, and some polysaccharides. We're going to talk about that in a minute, but this right here is called glycogen. Glycogen is a complex branched type of structure made of glucose monomers. So let's look at each one of these individually. Oh, here's another nice chart that has what the function is of starch. It stores glucose in plants. We eat the starch. Here it is, this amylose. It's a simple chain. We eat the starch and we digest it through hydrolysis in our digestive tract so we can get those glucose monomers for energy. Cellulose is another word for cellulose is fiber, and it forms some different types of structures, very complex structures, and that is for plant structure. It helps to make up the cell walls in plants, which keeps plants rigid. Plants don't have bones, so they need this cellulose. Glycogen, very complicated branched structure, is found in animal cells, in your liver, and in your muscles. It helps us to store glucose in our bodies. And then chitin, this is not chitin right here. I don't know why that's in there. But anyway, chitin, well, maybe that is chitin. Yeah, chitin's kind of branched too, is a found in the exoskeleton of insects. Okay, so let's look at each one at a time. So we already said that starch is officially known as amylose. When plants make sugars during photosynthesis, we eat those plants and then we break down the starch into sugar to get energy. Cellulose, like I said, is a complex structure that is found in cell walls. Here is a big microscopic view of a plant cell wall. You can see these cellulose fibers, and that keeps the plant rigid. Now, fiber, you may have heard of fiber, okay? There's fiber in a lot of plant products that we eat. And the reason that we eat fiber is because we actually don't have the enzymes in our digestive system to break these bonds, these glycosidic bonds that make up fiber. So it just passes right through us. But the good news is that it is good for our digestion because it keeps our colon cleaned out, and you know that you're not getting enough fiber if your feces are very dark and very hard. Um, you're getting probably, I'm sorry, you're getting too much fiber if your feces are really dark and hard. You're not getting enough if they're really watery and loose. So you want them to be just nice and, comfortable. And that's why we eat fiber. When you eat broccoli, any type of vegetable, you're eating fiber. When you eat a lot of fruits, you're getting fiber. So that's a good thing. Glycogen, like we said, is stored in animals in the liver so that when we need energy quickly, our body will perform glycogenolysis, which is the breaking down of glycogen. So our liver will release glucose, uh, will break down the glycogen and release it for quick energy. When we have too much sugar in our bloodstream, we can store it through glycogenesis into our liver. And if we get even too much, if we get even too much of that, um, if we get too much sugar in our bloodstream or in our liver, then our bodies will move that glucose out of our liver and it'll be stored as fat. Chitin is the exoskeleton of many invertebrates, most invertebrates, actually. Uh, Invertebrates are, of course, animals without a backbone, so they need some way to keep everything inside. And this uh, carbohydrate, this polysaccharide, is what is on the outside of shellfish and insects and even mushrooms. Fungus have chitin in their cell walls to give them structure. So when you crunch a bug and it goes, that's chitin. All right, so what I'm going to do is I'm going to walk you through quickly a couple of tests that we're going to do, uh, that scientists do to check whether are these polysaccharides or these monosaccharides in substances. One of the main things I can think of is if you are a forensic pathologist or you are a coroner, you would do tests on the contents of the stomach of a victim, and you would see what they had eaten, and you could put together maybe where they had their last meal. So we're going to pretend that we're a forensic pathologist right now. And I'm going to actually demonstrate this in just a minute. But so we're going to demonstrate the presence of monosaccharides by adding Benedict's solution. So here's what Benedict's solution looks like when it's in a substance that we want to test for the presence of monosaccharides. This test is negative. This test is positive. And like I said, I'll be showing you that in just a minute. If we want to test for the presence of starch, you can see here in this diagram that the starch is sort of an amber golden brown color. When we drop the iodine onto the starchy substance, in this case it's looks like a piece of potato, it will turn blue black. So we know that that starch is present. All right, so those are the two tests for the presence of both monosaccharides for in Benedict's solution, tests for glucose monomers, and the iodine tests for starch, amylose. And then this is a nice flow chart, okay? Carbohydrates can be broken down into monosaccharides, glucose, fructose, galactose, disaccharides, sucrose, lactose, maltose, Don't worry about oligosaccharides, but polysaccharides would be starch, glycogen, cellulose, and you may want in your notes to draw one more arrow. I apologize, it's not on this. That would be chitin. And now you're done.